In the next few minutes, we are going to pull back the curtain on one of the most powerful technologies humanity has ever created. You will see how CRISPR is already changing the way we treat disease, how it could end entire pandemics before they begin, and how it might even resurrect animals that have been gone for thousands of years. But we will also confront the other side of the story, the experiments that could alter ecosystems permanently, the attempts to design human traits before birth, and the choices that have sparked global debates about what it means to be human at all. By the time you reach the end, you will know about a CRISPR experiment happening right now that could shift the direction of evolution itself. If it succeeds, it could save millions of lives. If it fails, it could trigger consequences we may never be able to reverse. This is not a tale of science alone. It is a story about power, about responsibility, and about the fine line between creation and destruction. And it is already underway. To understand why CRISPR is such a revolution, we need to step inside the code of life itself. Every living thing on Earth, from the smallest bacterium to the largest whale, is built from instructions written in DNA. These instructions are made up of just 4 chemical letters, repeated and rearranged into sequences that tell cells how to grow, how to function, how to survive. In humans, there are about 3 billion of these letters, packed into every single cell. For most of human history, that code was untouchable. We could read some of it, we could try to understand parts of it, but changing it? That was like trying to edit a book written in a language we barely understood, without the ability to erase a single letter. Earlier attempts to alter DNA were slow, imprecise, and expensive. Scientists could try to make a change, but often it would happen in the wrong place, or it would damage other parts of the genome. It was like performing surgery with a chainsaw. Then came CRISPR. At its core, CRISPR is a natural system found in bacteria. A clever defense mechanism, these microbes evolved to fight off viruses. The bacteria keep a kind of genetic mugshot of their enemies, storing bits of viral DNA inside their own genome. In 2012, scientists realized they could take this microbial defense system and reprogram it. Instead of targeting viruses, they could make it target any DNA sequence they chose. In any living organism, it is targeted, it is efficient, and compared to older methods, it is remarkably cheap. A CRISPR kit can cost less than a dinner for 2. That means it's no longer locked away in billion-dollar labs. It is in the hands of researchers all over the world. The speed of progress has been staggering. In just over a decade, CRISPR has gone from a curiosity in bacterial immunity to a tool that can edit the DNA of plants, animals, and humans. It has already been used to make crops more resilient, to treat rare genetic disorders, and to create animals that could help stop the spread of deadly diseases. For the first time, we have the ability to go into the source code of life and make deliberate changes. Not random mutations, not blind trial and error, but precise, intentional edits. And when you have a tool this powerful, the question stops being, can we do it? And becomes, what will we do with it? The moment CRISPR became a practical tool, the world of biology exploded with possibilities. It was as if someone had handed humanity the master key to the genetic vault. And suddenly, researchers everywhere were asking the same question. What if? What if we could remove the mutation that causes sickle cell anemia, not just in one patient, but in every future child that family has? What if we could make crops immune to pests without chemical pesticides? What if we could eliminate diseases that have killed millions for centuries? And so the experiments began. In medicine, CRISPR is already rewriting the rules. Researchers have used it to edit immune cells, turning them into precision weapons that can track down and destroy cancer. Patients with inherited blood disorders, once facing a lifetime of pain and hospital visits, are now walking out of clinical trials free from their disease. These are not theoretical successes. They are real people whose lives have changed because a handful of letters in their DNA were corrected. In agriculture, CRISPR is giving us the ability to build resilience into our food supply. Scientists have created rice varieties that can survive prolonged flooding, wheat that resists devastating fungal infections, and tomatoes that stay fresh for weeks without spoiling. These changes could feed more people with fewer resources, even in the face of climate change. Then there's conservation. perhaps the most ambitious arena of all. Imagine reintroducing species that human activity has pushed to the brink, or even beyond it. The woolly mammoth, extinct for thousands of years, is now part of a serious de-extinction project. Using CRISPR, scientists are inserting mammoth genes into the DNA of Asian elephants, aiming to create a hybrid that could one day walk the Arctic tundra again. Some projects are even more radical. In certain parts of the world, CRISPR is being used to design what's known as a gene drive, a genetic change that doesn't just stay in one organism, but spreads rapidly through an entire population. With this technique, mosquitoes could be engineered so that almost all their offspring are male. Without females, the species collapses, and along with it, the malaria parasite they carry. This is not just controlling a disease, it is rewriting the destiny of an entire species. But each of these breakthroughs carries its own set of questions. What happens to the predator that relied on those mosquitoes for food? What happens to the ecosystem if we bring back an extinct species that no longer fits the modern world? What happens when edits spread in ways we didn't intend? Still, the pace of progress is relentless. In less than a decade, CRISPR has moved from a laboratory curiosity to a global force touching medicine, food, and the natural world itself. And some of its most ambitious experiments are not being tested on plants or animals, but on humans. And one of those experiments, happening right now, could push CRISPR beyond anything we have ever imagined. Every powerful technology forces us to confront questions we've never faced before. But with CRISPR, those questions cut deeper than most. They reach into the very definition of life, of identity, of what it means to be human. The same tool that can cure disease can also be used to alter traits that have nothing to do with survival. Eye color, height, intelligence, athletic ability. The line between medicine and enhancement starts to blur, and suddenly we are not just preventing suffering, we are designing people. In 2018, that line was crossed. A scientist in China announced that he had edited the DNA of twin girls before they were born, claiming to make them resistant to HIV. It was the first time in history that CRISPR had been used to make a permanent, heritable change in a human being. The news sent shockwaves through the scientific community. Many saw it as a dangerous leap into uncharted territory, driven more by ambition than caution. The scientist was condemned, his work labeled reckless, and he was eventually imprisoned. But the genie was out of the bottle. If we can make changes that pass from parent to child, those changes could last forever. They could shape not just individuals, but entire generations. Once released into the human gene pool, there is no easy way to take them back. And it's not just about people. Gene drives, the same technology that could wipe out malaria, could also wipe out species that become targets for other reasons. Some scientists worry that CRISPR could be used as a biological weapon, not to spread a disease, but to subtly change the DNA of a population without their knowledge. Even in the safest hands, the risks are real. Ecosystems are complex, interconnected webs, and a single genetic change in one species can trigger cascading effects we can't predict. A fix for one problem could spark another we never saw coming. That's why some call this moment in history an ethical earthquake. The ground beneath our understanding of life is shifting, and the aftershocks will be felt for decades, maybe centuries. Every decision made now will echo forward, shaping futures we can't yet imagine. And yet, in labs across the world, the work continues. Because for many scientists, the potential to save millions of lives outweighs the fear of what might go wrong. But there is one CRISPR experiment happening right now that embodies both the hope and the danger in a single breathtaking act. If it works, it could change the future of evolution itself. And if it fails, we stand at a turning point in the history of life. For the first time, a single species has the power to change the course of evolution, not in theory, not in the distant future, but here and now. With CRISPR in our hands, we can choose to erase diseases that have tormented humanity for centuries. We can protect crops, restore damaged ecosystems, and even bring back pieces of the past we thought were gone forever. We can use this tool to safeguard life on Earth in ways nature alone never could. But the same technology can be used to push past boundaries we may later wish we had respected. We can design traits to match our preferences. We can eliminate entire species because they inconvenience us. We can make changes that ripple forward through generations, long after we are gone, into futures we will never see. Once those changes are set in motion, there is no rewind button, no second draft. That is the reality we now face. Evolution is no longer a distant, slow-moving force beyond our influence. It is something we can guide, accelerate, and reshape, for better or for worse. The fork in the road is here. Down one path, a world where CRISPR helps us build a healthier, more resilient planet. Down the other, a world where our ambition outpaces our wisdom, and we create problems far greater than the ones we set out to solve. And the choice is ours. If you enjoyed this video, please don't forget to subscribe. And if you want curious questions answered, have a look at some of our other videos.